Hello everyone, I am S.V Gaikwad. I welcome you all in the B.Sc paper 2nd, that is BO332 Cell and Molecular Biology. So today we are going to dealing with the topic 3rd, that is prokaryotic cell. So let's start this session. So in today's session we are going to discuss the definition of prokaryotes, then general characters of prokaryotes, forms of bacteria, types of bacteria, and some of the references. So let's start. Coming to the definition of prokaryotes, so the word prokaryote derived from Greek word that is pro means primitive and karyon means nucleus. It means the organisms which have primitive nucleus called as prokaryotic organisms. Now what is meaning of primitive nucleus? See, in eukaryotes such as plants and animals, there are cell organelles, then there is a well organized nucleus. Means there is a nuclear membrane, there is nucleolus, there is nucleoplasm. So such type of organized nucleus is called as true nucleus, and the nucleus in which these organelles, nucleoplasm, nucleolus, absent is called as primitive type of nucleus. So the prokaryotes contain primitive type of nucleus, that is they lack a well defined nucleus. Okay. So these are certain examples of prokaryotes. The well-known example of prokaryote is bacteria. So these are cocci, means spherical shaped bacteria, and these are bacilli, means rod shaped bacteria. Okay. So as compared to eukaryotes, that is plants and animals, prokaryotes possess relatively simple structure. Okay. Let's see the general characters of bacteria. Coming to the definition of prokaryotes or bacteria, they are unicellular prokaryotic microorganisms. Okay. Microorganisms means the organisms which we cannot see by our naked eyes. Okay. For example, bacteria are microscopic in nature. So, for the study of these bacteria, we have to take the microscope. Okay. So, such type of organisms which we can't see by our naked eyes are called as microorganisms. The word unicellular means they are made up of single cell, and prokaryotic means they contain primitive nucleus. So, bacteria are unicellular prokaryotic microorganisms. The second feature of bacteria is they occur almost everywhere. That means they may present in air, may present in water or soil and inside other organisms. And such organisms which are present everywhere called as omnipresent. Okay. So bacteria are omnipresent. Certain bacteria, they may survive at very high temperature or very high salt concentration. And such type of bacteria, they are called as archaebacteria or archaea. For example, certain archaea, they may be present in hot springs also. Okay? A teaspoon of soil may contain several hundred millions of bacteria. As we know, what is the million, what is the billion, etc. So, as I said, bacteria are present in soil also. So, a teaspoon of soil may contain several hundred millions of bacteria. At such extent, the bacteria are microscopic in nature, microorganisms. Okay, so mostly bacteria are heterotrophic in nature. Heterotrophic means they cannot prepare their food themselves. Okay, but some bacteria, they contain bacteriochlorophyll in them. So, the bacteria which have bacteriochlorophyll, they can prepare their food themselves. So, they are autotrophic. In this way, some bacteria, they are heterotrophic and some bacteria, they are autotrophic. Okay. Next feature is some bacteria, they are parasitic or saprophytic. The word parasite means the organisms which survive on another living organisms. They are called as parasitic. Adversely, some organisms, they survive on dead and decaying matter, and the organisms which grows on dead and decaying matter, they are called as saprophytic in nature. So, bacteria may be parasitic or saprophytic. Next feature is some bacteria, they may be aerobic or anaerobic. The word aerobic means organisms which require oxygen for their survival, called as aerobic, and bacteria or organisms which don't require oxygen for their survival are called as anaerobic in nature. So, bacteria, they may be aerobic or anaerobic. Next feature is typically bacteria ranges between 1 micron to 3 micron. Friends, in previous session we have seen different units of measurement, and as bacteria are microscopic in nature, so for the measurement of their cell, the unit is micron. Okay, so majority of bacteria ranges between 1 micron or micrometer to 3 micron. Okay, one of the important feature of bacteria is complex cell organelles. Organelles such as endoplasmic reticulum, mitochondria, then Golgi complex and chloroplast. These type of organelles are present in plants and animals, but in bacteria, these cell organelles are absent. Okay? And the well-known examples of prokaryotes are bacteria, cyanobacteria, and mycoplasma-like organisms. Okay? Cyanobacteria, they also called as blue-green algae, and mycoplasma-like organisms, shortly called as MLO. MLO, they causes a disease called little leaf of brinjal. Okay, so So these are certain general characters of prokaryotes. Let's move on the forms of bacteria or shapes of bacteria. So basically there are 4 forms of bacteria. One is cocci, second is bacilli, third is spirilla, and fourth is Vibrio. So let's see one by one. Cocci means the bacteria which are spherical in shape. Okay, so this is the figure of spherical shaped bacteria, that means cocci. Whenever there is a single bacterium, it is called as coccus, and when there are many bacteria which are spherical in shape, they are called as cocci. Okay, and the well-known example of coccus is Micrococcus equisens. Okay, so the second form is bacilli. Bacilli means rod-shaped bacteria. So this is the figure of bacilli. When there is a single rod-shaped bacteria, it is called as bacillus, and when there are many bacteria which are rod-shaped, called as bacilli. Okay, and the well-known example of bacilli is Salmonella typhosus. Salmonella typhosus is a bacterium which causes typhoid diseases. Okay, the third form is spirilla. Spirilla means spiral-shaped bacteria. If there is a single spiral-shaped bacterium, it is called as spirillum. And when there are many spiral-shaped bacteria, they are called as spirilla. Okay, and majority of spirilla, they are motile in nature, for which they have flagella for the locomotion. So, well-known example of spirillum is Treponema pallidum, which causes sexually transmitted diseases. Okay, so this is the figure of spirillum. And the last form of bacterium is Vibrio. Vibrio means curved comma-shaped bacteria. This is the figure of Vibrio, which have single flagellum. So the example of Vibrio is Vibrio cholerae. Vibrio cholerae causes cholera disease. Okay, so the basic forms of bacteria are 4: cocci, bacilli, spirilla, and Vibrio. Cocci means spherical, bacilli means rod-shaped, spirilla means spiral-shaped, and Vibrio means curved comma-shaped bacteria. So let's see what are different types of cocci. Okay, so cocci means spherical or round-shaped bacteria. So whenever there is a single coccus, it is called as micrococcus. Okay, or micrococci. When the cocci occur in pairs, they are called as diplococci. Diplo means two, cocci means spherical. So when spherical bacteria present in pairs, called as diplococci, and the example is Diplococcus pneumoniae. This bacteria causes pneumonia disease. Next is the tetracocci. Tetra means 4. So whenever cocci present in groups of 4, called as tetracocci. Then sarcinae. When cocci are present in cubical arrangement of 8 or more, then it is called as Sarcini, cubical arrangement. Next is the Staphylococci. So whenever cocci present in irregular bunch or clumps and it resemble like bunch of grapes, at that time called as Staphylococci, and the example is Staphylococcus aureus, and the last form is Streptococci. So whenever cocci are present in bead-like chain, it is called as Streptococci, and the example is Streptococcus pyogenes. Okay, so these are certain forms of cocci bacteria. So here diagrammatically the cocci are shown. Single coccus, when cocci present in pairs called as diplococci. Then there is a group of cocci, Staphylococci. There is a chain of cocci, Streptococci. Cubical arrangement, Sarcina, and finally cocci in 4 number called as tetrad. So this is arrangement of cocci. Let's move on the bacterial types on the basis of gram staining. So gram staining, it is a staining technique. Stain means coloring or dyeing. Okay, so whenever a coloring is done to non-living things, it is called as dyeing, and whenever coloring is done to the living things, it is called as staining. So in laboratories we can stain plants or bacteria. So bacterial cell, due to difference in their cell wall composition, they are characterized into 2 types such as gram positive and gram negative. So the gram staining technique was developed by scientist Hans Christian Gram. Okay, so it is a differential staining procedure which differentiates bacteria into 2 groups, gram positive and gram negative. So this differentiation is totally depends on cell wall composition of bacteria. Okay, let's study the procedure of gram staining. Okay, so in this staining procedure there are 2 stains. One is the crystal violet and second is the safranian. So these all used for staining purposes. Another used medium is iodine. Iodine acts as a mordant or fixative which fixes the dye. Okay, so it acts as a fixative agent. And the next is alcohol. Alcohol acts as a decolorizing agent. D means removing. Colorizing means color removing substance called as alcohol. So it includes 4 steps. In first step there is a application of crystal violet. Okay, so when there is a application of crystal violet, all bacteria are colored purple. Okay, all will colored purple. After that there is a application of iodine, and iodine acts as a mordant which fixes this purple colored stain to the bacteria. Okay, after that there is use of alcohol. So alcohol acts as a decolorizing agent. After application of alcohol, there are some bacteria which loses purple color. Okay? After application of alcohol, there are some bacteria which loses purple color. Finally, for visualization of the second type of bacteria which removes that color, purple color, in such case there is application of another stain, that is counter stain. We can use safranin. So after application of safranin, some of the bacteria they colored pink. So at the end of this gram staining procedure, we will see two types of bacteria. Some of the bacteria they will colored purple and some of the bacteria they will pink in color. So the bacteria which are purple in color, they are gram positive bacteria, and the bacteria which are pink in color, they are gram negative bacteria. So finally we got two types of bacteria. Totally depends on cell wall composition. Okay, so this is all about the staining procedure, the general characters of bacteria, then forms of bacteria. So friends, these are certain references which I use for this session. So friends, this is all about today's session, and we will meet in the next session with the new topic of cell biology, that is ultrastructure of prokaryotic cell. Thank you very much.